Hello and welcome to Chapter 22, The Origin of Species. I'm Dr. Chris Whips. Let's get started. So Darwin, in his many observations, had described this, amazing diversity of life on the planet and thought of speciation to him as kind of this mystery of mysteries. It's like, where do new species come from? And really, that's what this chapter is about. We'll see many examples of how new species might originate. But speciation is also an interesting link between microevolution, which was what we've been talking about before, where we're looking at allele frequencies and populations are changing over time. It's often very sort of easy to digest. It's very quantitative. We can see change. Evolution is happening. We can quantify it. But then how does that get you to macroevolution, longer processes over time where new species arise? And that's macroevolution. So the core of macroevolution is speciation. So speciation is that bridge between. And here's just a simple example. Here we have a single population. Maybe they are separated. And then you'll start to see changes, thinking a microevolutionary perspective. Maybe these organisms we'd still consider the same species at this point, but given enough time potentially or enough genetic changes, we see that divergence. And so there's that foundation of macroevolution. And here we see the populations come back in contact, but they no longer interbreed. And at that point, we'd consider them distinct species. So this maybe sounds like a trivial question, but what is a species? It is a fundamental question and sometimes not as easy to answer as it may sound. We look at these organisms like in these pictures here, and we say, oh, well, here's a mussel and well, here's a barnacle too. These things look different. Those things are different species. But there are many ways that we can define a species. And one of the ways that we use is called the biological species concept. So we use different species concepts to define species. This has to do with the ability to interbreed. So we can specifically define the BSC, biological species concept, as groups actually are potentially interbreeding with natural populations, which are reproductively isolated from other such groups. And then this comes back to microevolution, the idea that there's gene flow, say, between populations, that we would consider those the same species. If there's no gene flow, then we would typically consider those different species. But again, it's the potential, right? Maybe there's not gene flow right now, but if you put them together, could they interbreed? And that is the core of the biological species concept. And so this is great for some organisms, but you can probably imagine this is not so great for if you're, say, studying fossils. You have an extinct species, you're never going to know if that can interbreed with another similar looking group. Organisms that asexually reproduce, not useful there because biological species concept's all about sexual reproduction. The emphasis is gene flow, and there may be other ways to look at genetic exchange in these other kinds of organisms, certainly not in fossils. And so we really need some other tools. So these other species concepts typically emphasize looking at similarities between species in different ways. And this first one is called the morphological species concept. And this one, I think, is maybe the most straightforward. And probably this was the earliest species concept. You look at something and you say, this one looks different from that one, ergo, those are different species. Maybe some issues with that, of course, it may rely on subjective criteria. You might imagine certain organisms where there's dimorphism between individuals within the species, a male and female, for example. Peacocks, for example, males look strikingly different from the females, but we don't consider those different species. So you have to look at potentially a lot of characters all together, and maybe tie in some of these other species concepts where possible. The ecological species concept defines species in terms of their ecological niche. So you're looking at what an organism does, where it lives, how it interacts with other components in its environment, other organisms. And this could be applicable to sexual or asexual species. Maybe not so much with fossils, although it depends on what you might be finding in those fossils or around those same fossils. This does emphasize a role of disruptive selection. Now, this is something that we've already talked about, and we'll see here again in speciation, where you have maybe a population that you have two distinct groups that are utilizing different resources, like these birds with the seed size here. Big birds with or big seeds can be eaten by birds with big beaks. Little seeds can be eaten by birds with little beaks. And there's, say, no intermediate size seeds. As you start to see these populations diverge, and potentially this is that foundation of speciation, and it really relies on these different ecological niches. And so you can apply this ecological species concept to similar situations, or bona fide distinct species. You'll see those ecological differences. Another way to define species is through phylogenetics. We've talked a lot about phylogenetic trees before. Here we can, say, generate DNA sequences, or potentially you could use morphological characters in a phylogenetic tree. This could be used for extinct species. If you're using genetics, that's species that still exist, extant species. And you're looking for groupings, right? So here's an example here, and this is some work I did many years ago. But these organisms that are parasites of fish here, we have this cluster of this one group, Cudoa thersites. We have 3 different samples from different places in the world, but they really tightly cluster together. And so we would consider this one species. I will also say we can't really apply a biological species concept to these particular organisms. Morphology is okay. We can use that as well here. And what we found here, there's this sister species. This one's called Cudoa mini thersites. It is slightly morphologically different, but also phylogenetically distinct. Here's a distinct branch off to the side of Cudoa thersites here. So these guys look similar. There are some differences, but there's also this phylogenetic difference. So this can be very useful. And this also emphasizes that point of potentially applying multiple species concepts to the situation that you're investigating. All right, so why don't you try to do that? Apply multiple species concepts and answer this question. All right, so yeah, if we're looking to place dogs and wolves into the same species, that biological, because they can interbreed, and phylogenetic, because of very recent common ancestry, those would be very applicable here. So let's circle back to the biological species concept, because here's where we can really see how speciation might occur, whether we can apply this species concept to everything or not. This is a great way to look at how it might work. And the BSC really focuses on reproductive barriers, because if you think about it, that's really how the biological species concept is defined. It's foundationally about reproduction. And so if reproductive barriers are occurring, that would suggest that we're starting to see divergence between species. And we'll talk about all of these in due time. But let's first start with, well, why might we see reproductive isolation? You can think of these as barriers to gene flow, right? Think back to microevolution. So if we don't have gene flow, we would start to see potentially divergence over time. And a barrier to gene flow might be small, like this little stream here is a big barrier to gene flow, say for these little insects here. Maybe that barrier is huge. Here is the Grand Canyon. You've got 2 different squirrels on either side of this Grand Canyon, 2 different species of squirrels, but they're in the same genus. And this would cause a geographic isolation. When populations are geographically isolated and then those groups diverge from one another to the point where we consider them different species, this is called allopatric speciation. Allo basically means different. Patric means country. So think of it like different place. So they're separated. And it kind of makes sense. Like, well, if they're separated, there's no gene flow between them. They're just by chance over time, they will diverge. And that time could be very short or it could be very long. It depends. And you can see this I like this cartoon here showing, well, we have a continuous population up here at the top. There's some sort of split that causes a divergence of population A and B here. And then again, over time, you'd see mutations randomly that would occur in these different populations. They're not going to be the same mutations in population A and B. And if you were to bring these back together at different points early on, these organisms may still be able to interbreed. And we'd say, well, okay, they're still the same biological species, even though we're starting to see some genetic differences, definitely different populations, but they can still interbreed. But as more time passes, here we're seeing this X here showing that at some point, these populations now can no longer interbreed because of accumulated mutations, evolution within those populations, and we now have what we consider distinct species. And so we can think about this allopatric speciation as being driven by vicariance. So think back to biogeography that we talked about. Vicariance is a splitting of a population. And the example here would be the Isthmus of Panama, the rise of that about 3 million years ago. And this was a slow process. So this rise was not just 3 million years ago. It was 20 million to 3 million years ago, where you started to see rising under the sea. So it's getting shallower and shallower, and then ultimately completely cut off, separating the Caribbean and the Pacific. And so a consequence of that is splitting of these marine organisms on either side of this Isthmus of Panama. It creates a barrier to gene flow that cannot be overcome by these organisms. And what do we see as a result? Well, we see these different kind of snapping shrimp, and you have a wide range of varieties of these species. And if we look phylogenetically, what do we see? These lineages of snapping shrimp, where you have one branch where you have the Pacific, this is species number 6, for example, up here. You have a Pacific lineage and a Caribbean lineage. This shows that there was, what do we got here? 7 different, say, snapping shrimp species before the Isthmus of Panama rose and created a blockage to gene flow. And when that happened, each of those 7 would split. So, species number 3, you've got a Pacific version and a Caribbean version. Species number 5, you've got a Pacific version and a Caribbean version, and so on. So in all these lineages, we see a speciation event between, of species 6 and species 3, etc. All right, so think about this. And remember, keep in mind that the Isthmus of Panama, as it arose, say it started out as flat. Let's just imagine that. And it's going to get taller and taller over time to the point where it finally is breaking the surface and creates a complete barrier to all of these. Now, in this question, we have these snapping shrimp that occur at different depths. So you have to think these are going to get cut off from one another on either side of this isthmus at different times. All right, so what do you think, or what would be the snapping shrimp most closely related to those that live in this habitat in A4? All right, yeah, so it would be B4. Go back to that phylogenetic tree. This would be the sister group, when the isthmus was down here, these were connected populations. So this would have been one species at level 4, or maybe there's multiple species, but we'll just say it's one for the purposes of description. And then as that rose, as soon as the isthmus was high enough or shallow enough, I guess you can think of it like that, to cut off the side A and B, at that point, those organisms would begin to diverge. So thinking back again to biogeography, another way that you can have isolation would be through dispersal. So here's an example here of what's called peripatric speciation. Peri kind of means around or near. And this is still allopatric speciation, but we have this collection of bird species in New Guinea, and we have these distinct populations as shown highlighted here. Some are on islands, some are on the mainland, but separated by geographic distance. And this is a result of dispersal of these birds from one place to another and then subsequent speciation. Now, you don't necessarily need a huge river or a mountain range to create that geographic isolation, but just simply distance can be a reproductive barrier, right? So if you look at the degree of reproductive isolation in geographic distance in this whatever random species we're using just as an example, what we see is as the distance increases, the degree of reproductive isolation increases. Now, obviously, that's going to be different for different kinds of organisms. For those little beetles, maybe a very small geographic distance, it makes for a big barrier. For animals that can fly, birds, a big distance might have to be thousands of kilometers, not hundreds, to create just a raw distance being that barrier. Now, as a disease biologist and parasitologist, one of the things that we see in these fields is something called co-speciation, where you have these parasites like these chewing lice, for example, that they're very tightly associated with their host species. And when the hosts diverge phylogenetically, we actually see that line up with the divergence of these lice species. Because, if the hosts are speciating and separating, they're no longer overlapping and interbreeding. They're also not transmitting their lice from one to another. And so then the lice diverge too. And so from the perspective of the lice, the hosts can almost be thought of as, their habitat, the place they live. And so when the hosts separate, that's almost like allopatric speciation, which is really cool. All right, so here's a question to consider, and choose the best answer. All right, yeah, the best answer here is kind of the it depends answer, right? So whether or not this is going to have a big impact on a particular species depends on that species' mobility. For a bird, for example, this is probably not that big of a deal. But for a little rodent that lives in the forest, this is probably a huge deal. It creates a massive barrier. All right, so this concept of geographic isolation, allopatric speciation, hopefully makes sense, because you can imagine a mountain range sort of blocking the little mice from moving from one side to another, or the Grand Canyon with the squirrels. Now, is geographic isolation needed? Does it always have to happen? And the answer is no. We can see where there are examples where you have species or populations that overlap, but speciation still occurs there. And this is called sympatric speciation. Sympatry means same country. And so we see speciation happening in the same place. So fundamentally, you still need to have barriers to gene flow. But what would stop it, right? If it's not the mountain range, that seems the obvious one. What could cause that? And there's a few things that might cause that. One's called polyploidy, habitat differentiation, and sexual selection. And you can envision how this might happen here in sympatry within the population. So we haven't separated it. We have a speciation event occurring. So let's talk about that polyploidy first. Now, when we talked about genetics, we talked about ploidy, and polyploidy is just where you end up with extra sets of chromosomes. Typically, this is an accident during cell division, and this might actually lead to speciation. Most of the time where you have a mistake like this occur, it's probably has a negative effect, but it could have this impact where you have speciation occur. Now, an autopolyploid, and think, when you see auto, think self. This would be an individual in a species that has more than two sets of chromosomes derived from that same species. So auto, self, sort of think within a species. And one particular kind that's really important here are the tetraploids. So think of this as a doubling of the diploid state. So everything has been doubled and you have tetraploid organisms. Now, if you have the mating between a tetraploid and a diploid organism, you end up with a triploid, 3N. Triploids typically have reduced fertility, so here's a hybrid version between these two different organisms that is doesn't do very well. However, a tetraploid that can either self-pollination, again, we're talking mostly about plants here, or if by chance mates with other tetraploids, this will produce a tetraploid offspring, right? So, and those individuals are potentially equally fit or certainly fertile relative to the parental generation. Now, another way to get a polyploid is the example here where we have an allo polyploid. Now, remember, like allopatry, allo means different, right? So you're taking gametes from two different species, those fuse, creating a hybrid organism. And as we see in the example here, so we've got these gametes coming together, you end up with a sterile hybrid zygote. So this organism is sterile. So, okay, that doesn't sound great. But again, some mistake occurring during mitosis or meiosis, you end up with this doubling of the chromosomes. Now you have this diploid organism. It has the whole set of chromosomes that were from both parents at this point, and now can potentially self or interbreed with other similar hybrids. All right, so here's a question to answer. All right, yeah, so once you have these polyploid organisms, they then can't typically reproduce with those parental species. And of course, if they can't reproduce with those parental species, then think biological species concept, we would consider them distinct species. Thus, speciation has occurred. All right, so another kind of sympatric speciation that can occur is called habitat differentiation. And this really connects back to that ecological species concept and disruptive selection. And we've actually seen this example before when we talked about disruptive selection, where you have flies that feed on apples and hawthorns. Before apples were introduced into North America, these flies only fed on hawthorns. But now you really have two different populations or potentially species here, that these flies occur in the same geographic places, but because they're utilizing a different resource, a different ecological niche, they have evolved a different phenology. So you see the timing of when these organisms are present. For the ones that feed on apples, those peak in the beginning of August, versus those that feed on the hawthorns, those peak in September. Therefore, when these guys are breeding, they're breeding at different times, and then obviously less likely to interact and breed, and that has created this reproductive barrier even within the same geographic location. All right, the other way that we can see sympatric speciation happen is through sexual selection. So here we have mate choice really driving differentiation between species. And a great example of this is cichlid fishes in the Great Lakes of Africa, specifically Lake Victoria. That's the one we'll focus on. It's really interesting because you have what is a very recent radiation of these fish. So if we look at these different lakes and these lineages over time, if we look at these lakes like Malawi and Lake Victoria, you have really shallow lineages, meaning there's a lot of recent divergence of these cichlid fishes, and it's primarily driven by sexual selection. And so how does that work? Or what is our evidence for this? Well, we've got these two different cichlid species, and females under normal conditions, so normal light conditions, they would choose males of their own species to mate with. So they're recognizing the coloration of the fish, and they say, okay, that male's in my species, that's who I'm going to mate with. But if you experimentally put these fish under a monochromatic light, so you're housing them in these conditions, the females will mate indiscriminately with males, whether it's what apparently is one species or another, and they can mate. And so that would suggest, well, are they then a different species at all from the biological species concept? So what's happening here is we're seeing under normal conditions, mate choice is driving the isolation between these different species, or what maybe border on different species. So whatever is initially driving the separation of species, whether it's allopatry or sympatry in those different examples there, we see these initial steps being reinforced by these barriers to gene flow. And again, we're focusing here on that biological species concept. So we'll first look at the pre-zygotic barriers. So this means before fertilization is happening, pre-zygotic. So no zygote is formed, meaning you don't get a fertilization event. But there are also barriers that are post-zygotic. So think about hybrids. So sometimes a fertilization event can occur, but it doesn't necessarily result in a viable fertile offspring. If it does, that would suggest that you're not dealing with different species and there isn't a barrier. So we're getting the barriers are getting lower and lower as we move down from these pre-zygotic to post-zygotic barriers. So let's talk about the pre-zygotic barriers first. The first one is ecological or habitat isolation, which we've already talked about several times, where you have different populations initially utilizing a different niche. And then this is, driven by disruptive selection, and then we start to see speciation occurring. So if, these water-dwelling snakes live in water, these guys live on land, they're not going to have occasion to bump into one another and mate. Now that, if you take the example of lions and tigers, these are ecologically isolated and geographically isolated. But if you put them together in, say, a zoo, you can actually get hybrids of lions and tigers, or tigers and lions, male, female, depending on which you have. You either get a tigon or a liger. And so these organisms can interbreed, but they don't, and unless they're in like the zoo situation here. Now, some organisms do breed in the wild and create hybrids. The example here are grizzly bears and polar bears. They don't typically interbreed, but they can, and you end up with what's called a growler bear, which is a hybrid of a grizzly and a polar bear. So these are generally ecologically separated and don't interbreed, but they can. And so we're sort of stepping from pre-zygotic to post-zygotic. But again, most of the time, this is a pre-zygotic barrier. Behavioral isolation. So this is, as it sounds, if an organism behaves in a certain way, it's not going to have or is not going to have a mating opportunity with organisms in that same population or a different population. And so the example we use here is these blue-footed boobies. They have a certain mating dance that they have to do. They have to exhibit this certain behavior that allows them to mate. And if you don't exhibit that behavior, maybe because you're a different species or a different population, those mating opportunities are gone. So this would be a behavioral isolation. Temporal isolation. Now, I will emphasize temporal means time. Sometimes people, if you hadn't heard that before, you might think of temperature. There's nothing to do with temperature. It's all about time. The example here would be these spotted skunks. So they have ranges that overlap. So there's that potential to interbreed, right? They do at least overlap. But these organisms, the eastern spotted skunks, they mate in winter, whereas the western spotted skunk, they mate in summer. So they're mating at completely different times. Therefore, because of that, this is temporal isolation. You're not mating at the same time, you're not going to have offspring. Another example is mechanical isolation. Now we're kind of getting it to the point where a mating attempt might be attempted, mating might be attempted, but it doesn't work. So we have these two different snails with shells that turn in opposite directions, and this actually prevents the snails from physically mating. So this can't happen. So it's just mechanically the mating event can't work, even if it's attempted. As we move further along here, there's gametic isolation. So maybe a mating attempt has occurred, but the gametes can't fuse. Or in this example here, there's a picture of urchins here. So urchins and some other kinds of marine organisms like them, they broadcast spawn. So they'll just, send their gametes out into the water here. And so those might all be present. So there's the potential that they could fuse, but they don't. Only the right sperm will fuse with the right egg, so from the same species. So that's gametic isolation. All right, here's another question for you to try. All right, this one might sound a little bit like gametic isolation, but it's really more a mechanical process here. So the pollen grains cannot make that pollen tube, and therefore just physically those gametes cannot come in contact. All right, so next we'll talk about the post-zygotic barriers to gene flow, and then we'll talk about hybrids and what we see and how that gets us close sometimes to speciation in the next part. We'll see you then.